Hello and welcome to this video where we're going to be looking at our last macromolecule, which is proteins, and a specific type of protein called enzymes. So as you can see here, we've already talked about carbs, lipids, and nucleic acids, so now it is time to talk about our last macromolecule, which is proteins. So to start, some basic facts about proteins. So proteins are made up of 4 elements mainly, and then a 5th element sometimes. So you can see all proteins have carbon, hydrogen, oxygen, and nitrogen, and some proteins have sulfur in them. But the monomer of a protein is called an amino acid, and this is abbreviated AA. There are 20 different types of amino acids that make up proteins in humans, and the polymer of a protein is called a polypeptide. Now, I want you to imagine if you had a long chain of amino acids, so a long chain of monomers, that would be the polymer, a polypeptide. However, if this long chain folded onto itself to make a 3-dimensional shape, then we would call it a protein. So a protein is a folded polypeptide. So let's look a little bit more carefully at the monomer of a protein. So remember we said that the monomer of a protein is called an amino acid, and every amino acid has the same structure. So if we look at this picture over here, every amino acid has a carbon in the center, and this carbon is bonded to 4 things. It's bonded to a hydrogen atom, it's bonded to a amino group, which we already know, it's bonded to a carboxyl group, and then the 4th group is called the R group, or the variable group. And the R is not an element. R just stands for the rest of the molecule. And this group will vary depending on which of the 20 amino acids we're looking at. So let's look at some examples here. So you can see here I have 3 different amino acids. So you can say each one has an amino group, each one has a hydrogen atom, and each one has a carboxyl group. But for each amino acid, the R group varies. So you can see each one has a different R group in the yellow box. And it turns out the R group, or the variable group, is what gives each amino acid its specific properties. So for example, the amino acid leucine has a hydrophobic R group, which means this R group, if a protein has a lot of leucine amino acids, it would be nonpolar or hydrophobic. Contrastly, if I have the amino acid serine or aspartic acid, they have hydrophilic R groups. So you can imagine if I had a protein with lots of these amino acids, it might make the protein more polar, for example. So just like with any other macromolecule, if I want to build a polymer, I have to link the monomers, or amino acids in this case, together via dehydration synthesis. And specifically, if I do dehydration synthesis with 2 amino acids, I would remove the water here, would end up with this bond forming, and the bond between 2 amino acids we call a peptide bond. Now, I want you to think back to carbs. Remember, if I linked 2 monosaccharides together, I got a disaccharide? This is kind of similar in proteins as well. If I link 2 amino acids together, I get what's called a dipeptide. That's the polymer that has 2 amino acids. Okay, so here's another example of what we just looked at. You can see here's my 2 amino acids right here and right here. I'm doing dehydration synthesis to remove the water, and then I link those 2 together to form a peptide bond right there, and here's my dipeptide. Similarly, if I link 3 or more together, right, so here I happen to be linking 4 amino acids together by doing dehydration synthesis 3 times. You can see I'm removing my 3 waters. If I have 3 or more amino acids together, that's called a polypeptide, just like we had a polysaccharide with carbohydrates. Okay. So we said earlier that polypeptides is the name of the molecule we get when we link many amino acids together. However, polypeptides generally form a 3D structure. They fold on themselves, and that folded 3D structure gives us a protein. So over here, I have several different pictures of proteins. So you can see these long chains, these are supposed to represent the chains of amino acids. So these long chains represent the polypeptides. And you can see each different chain has folded on itself into a different shape. You can see each protein has a different shape. And that's really important because it turns out the shape of a protein is vital. It's super important for its job, for its function. So I'm going to give you an example. So for example, this protein right here, lysozyme, it acts to break down things in the cell. Specifically, this protein will kill bacteria. And the reason its shape is so important for its function, because you can kind of imagine, maybe perhaps the thing that it breaks down fits into this little space right here. So if it didn't have this particular shape, it wouldn't be able to bind onto the thing that it's going to break down, and it wouldn't be able to do its job. So proteins' jobs are dictated by their shape. Okay, so remember, a protein, we just said, is a polypeptide folded into a unique shape. And this shape is essential for its function. The shape determines its function. However, sometimes it's possible for a protein to lose its shape. And if a protein loses its shape, that's called denaturing. And if a protein denatures, if it loses its shape, it can't do its function anymore. So let's look at an example of that. So you can hear, here I have some protein and it has some shape, and its shape allows it to do its job. And if something were to happen to this protein, like maybe it got too hot or maybe certain chemicals were applied to it, it would unfold back into its polypeptide form. And because it now has unfolded and doesn't have its shape, it's not able to do its job. So we know that proteins fold, polypeptides fold on themselves into proteins, but kind of the question is, why would a protein fold? So I want you to imagine, remember we said in the last video that the order of nucleotides in DNA, right? Remember when we were talking about nucleic acids? So the order of nucleotides in a gene in DNA, remember we said that DNA codes for proteins. So the sequence of nucleotides in DNA dictates which amino acids we put together to build a certain protein. So you can kind of imagine if I had a lot of positively charged amino acids, or a lot of hydrophobic amino acids, or a lot of hydrophilic amino acids, the protein would fold differently depending on which sequence of amino acids were put into the protein. So whatever amino acids the DNA codes for, they interact with one another by either attracting one another or repelling one another, and this causes the polypeptide to fold into a protein. And you can kind of imagine, if I change one amino acid in a protein, maybe I add a polar amino acid, but there used to be a non-polar amino acid, that would completely change how the protein folds. So basically, the types of amino acids that are in a protein will dictate how the protein folds. So there are many different ways that amino acids can interact to cause them to fold together, to cause the protein to fold. So one way they can interact, so here I have two amino acids, so here's one, and here's the second. Sorry, I'm a slow drawer. Okay. So one way two amino acids can interact is we can get a hydrogen bond can form between the carboxyl group on one amino acid and the amino group on the other. That can form a little hydrogen bond there. And you can kind of imagine each of these circles on this picture here represent a amino acid. And the blue ones, like this amino acid right here and this amino acid right here, are hydrogen bonding. And because they're hydrogen bonding, and because these two are hydrogen bonding and these two are hydrogen bonding, it causes this polypeptide chain to start to fold on itself, right? So these hydrogen bonds can give the first folds that we see in a polypeptide to build a protein. So another way amino acids interact to cause a polypeptide to fold into a protein is that the R groups can interact. So remember before we said that we can have negatively charged R groups, positively charged R groups, hydrophilic R groups, hydrophobic R groups. So you can imagine, all the hydrophilic R groups, those amino acids are going to want to be on the outside of the folded protein near the water, right? all the hydrophobic R groups are going to want to be in the middle of the protein, away from any water that's in the solution. If you have a positive R group, it might be attracted to a negative R group, and all these different interactions cause proteins to fold in different ways. So the endless different combinations of amino acids can result in endless different ways a protein can fold, which can lead to lots of different protein shapes and thus lots of different protein functions. And then finally, the third way that amino acids can interact is, I want you to imagine that I have multiple folded polypeptides. So maybe here I have one polypeptide chain that's folded, and I had a second one over here that's folded. Sometimes these two different polypeptides can attract to each other to create a larger, like, mega protein. So only certain proteins do this, but more complex proteins, like things like hemoglobin and collagen, they reach this level of folding. So, to kind of summarize, the reason we care about how proteins fold is because a protein's shape, which it gets from folding, is vital for its function. So I'm going to give you one more example. So what I have here here's my little folded protein. So here's my chain of amino acids. And each of these little colored spheres represents a different R group, okay? So you can see maybe all of these R groups are, and I'm making this up, and maybe they're all hydrophilic, so they're all on the outside of the protein. And it turns out they're arranged in such a way that the molecule that they're going to interact with, like maybe their job is to break down this molecule, and they're arranged in such a way that they can bond with this molecule perfectly. Now, if this protein was unfolded or folded a different way, these R groups wouldn't be arranged in such a way that would make it easy for them to bind to this thing that they're going to break down. So therefore, if it didn't have the right shape, it wouldn't be able to break down this molecule. In other words, it wouldn't be able to perform its function. Okay, so the next thing I want to talk about is what happens when we eat protein. So now we know all about protein shape and why shape is important to function. But when you eat a protein, the first thing that happens is, just like with any other macromolecule, is that polymer, the protein, gets broken down into its monomers, into amino acids. First, the first thing your cells might do, if your cells need new proteins to do certain jobs in the cell, it will use those amino acids that it got to build its own polypeptides and its own proteins. And then any extra amino acids, any excess ones that are not needed to build proteins for the cell, are deaminated. And what this means, if I have my amino acid right here, right? Sorry, this takes me a while to draw this. It means if an amino acid is deaminated, it means the amino group, the NH2, is removed. So what this does is it produces two things. This half of the molecule, it turns out, can be converted to something that kind of looks like glucose. So that means the body can use that half that I just put a box around as a glucose-like substance and can use it to make energy in cellular respiration. So when I say it's used in respiration, I mean that it's used for energy because it kind of looks like glucose. Then the other half, the amino group, that is what's called nitrogenous waste. Nitrogenous meaning nitrogen containing. And this, you don't want this hanging out in the body because this can cause a lot of damage if it's left in the body. So we get rid of it via a molecule called urea, which is the main component of urine. So really the reason we urinate is to get rid of this, this waste molecule from breaking down proteins. Okay. So let's talk, we said that protein shape are essential for their functions. So let's talk about what some of the functions of proteins can be. So it turns out proteins do most of the work in your cells. Proteins do almost everything. So let's talk about what these jobs might be. So some proteins are called transport proteins, which means they transport things around the body. The main example that you should know is hemoglobin. So this is the protein that carries oxygen and carbon dioxide in red blood cells. So it carries oxygen through your blood system. Another job that proteins can have, and this is the one that usually you think of when you think of proteins, is proteins can be contractile. And what that means is that they can let your muscles contract and extend. So they allow you to move your muscles. So the main two proteins that do this are actin and myosin, okay? A third job that proteins could have is they act as the defense system in your body, defensive proteins. So an example here are antibodies. And antibodies are proteins that recognize things that are foreign, things that shouldn't be in the body, like viruses and bacteria, and they attack them and get rid of them. So the fourth type of protein we can have is a protein that provides structure. So its job would be to provide structure for the cell. So these things might be a protein called elastin, for example. That's the protein that gives your skin its elastic structure. Proteins like keratin and collagen are proteins that are found in your hair and your nails, and they're what give your hair and your nails structure. So a fifth job proteins could have is they can send and receive signals, signaling proteins. So we already know some of these. So some things like hormones and the receptors. So receptors are proteins that receive the signals that hormones send. So this might be a little confusing because we said in an earlier video that steroids, which are a lipid, are hormones. And it turns out half the hormones in your body are steroids, so half are lipids, and the other half are protein hormones. So kind of half of hormones are proteins, and then the other half of the molecules are a lipid. And some protein hormones include things like insulin, which signals your body to store glucose, glucagon, which we learned does the opposite, and growth hormone, which is a protein that signals your body to grow. Proteins can also store energy. They're storage proteins. So I want you to think about alpha-bumin, which is a protein that you know. It's the protein in egg whites. That's the reason why eggs have so much energy is because the egg, right, is storing a lot of energy. So if it gets fertilized and grows into a baby chicken, right, there needs to be energy to feed that chicken. So alpha-bumin is the protein that stores that energy in an egg. And then finally, the most important function of of proteins are enzymes. And enzymes are proteins that speed up chemical reactions. They make chemical reactions happen faster. So for example, the enzyme lactase speeds up the chemical reaction that breaks down lactose, right, the disaccharide. A lot of enzymes end in ASE. So if you see ASE, we're probably talking about an enzymatic protein. Now, that's going to be it for this video, and then in the next video, we're going to talk quite a bit more about these proteins called enzymes.